Hi everyone, my name is Jessica and today I'll be sharing with you my poster about investigating the ability of Serratia marcescens to degrade the insecticide diazinon in soil. Pesticides are essential tools in modern agriculture for controlling pests and protecting crops, but their long-term use can lead to soil contamination and disruption of microbial communities. Pesticides are chemicals used to control pest insects around the home and field crops. They contain both active and inactive ingredients, which are classified as conventional, biopesticide, and antimicrobial. Pesticides repel and prevent pests on plants and humans and can be detrimental to organisms that encounter this chemical. Serratia marcescens is an opportunistic gram-negative bacterium known for its ability to thrive in diverse environments. Serratia marcescens is a facultative anaerobe, which allows it to thrive in both oxygen-rich and oxygen-poor environments. It is capable of degrading various organic compounds, including pesticides, due to its ability to utilize a wide range of carbon sources and adapt to varying conditions. Bioremediation is a process in which a microorganism degrades environmental pollutants. Degradation relies on specific factors, including microbial communities, soil fertility, and environmental factors. I hypothesized that Serratia marcescens will degrade the insecticide diazinon and increase the population growth over time. This study aimed to measure the level of degradation of diazinon in soil following exposure to Serratia marcescens and to investigate changes in bacterial growth during pesticide degradation. The study's experimental design involved introducing Serratia marcescens into sterilized soil samples treated with different concentrations of the insecticide diazinon and monitoring bacterial growth and pesticide degradation over a 14-day period. Prior to the experiment, gram staining was conducted to ensure proper bacteria were being used. The zone of inhibition assay was conducted to determine the concentration of pesticide to be used during the experiment that wouldn't kill off the bacteria. The concentrations used for this assay were 0 parts per million, so just acetonitrile, 5 parts per million, 10 parts per million, and 25 parts per million of diazinon. The concentrations that were determined to be used for the experiment were 5 parts per million and 10 parts per million. Next, the soil was sieved, then sterilized in the autoclave 3 times. The 2 chosen diazinon concentrations were used to inoculate the soil with the bacterial culture. A vehicle control of bacteria and acetonitrile, abiotic controls containing the diazinon standards, and 2 treatment groups containing the diazinon treatments with bacterial were run. The soil was transferred into seeding pots, and samples were taken on day 0, day 4, day 7, day 11, and day 14. The samples were taken in triplicate and stored in glass vials at -20 degrees Celsius until analyzed. For each sampling day, pesticide concentration was measured using gas chromatography and an HP5 column. The stored soil samples were rehydrated with water and centrifuged. The supernatant was mixed with acetonitrile and 1 package of Ketcher's salt, then centrifuged again. The supernatant was transferred to a GC vial, and pesticide concentration was measured. On each sampling day, bacterial colonies were counted using the compact dry disc method. The soil was rehydrated with PBS and centrifuged. The supernatant was used to create dilutions to be plated. The sample was plated neat, 1 to 100, 1 to 1,000, and 1 to 10,000 fold in PBS. The plates were incubated for 48 hours, then the colonies were counted, and the total colony-forming units per gram of soil was determined. After thorough investigation, the study found no evidence that Serratia marcescens effectively degraded diazinon in the soil samples. However, despite the lack of direct diazinon degradation, the study sheds light on the intricate relationship between pesticides and soil microorganisms. Furthermore, bacterial plate counts showed an interesting trend, an initial population decrease followed by significant recovery. This resilience suggests that Serratia marcescens possesses the ability to withstand and adapt to the stress induced by the presence of diazinon. It's crucial to note that errors in the study, including improper mixing and inoculation, may have affected the results. There are many factors that could influence degradation rates, such as temperature, pH, and soil type. Moving forward, exploring these conditions would help to determine external factors that could impact pesticide degradation rates in soil. Future investigations can look into how soil health is affected in the presence of pesticides, as well as comparing the performance of the bacteria to other pesticides. An extended time frame would also be interesting to see, as many articles I have read conducted similar research for upwards of 1 year. In conclusion, while the study's findings are mixed, they highlight the potential of Serratia marcescens as a bioremediation tool for organophosphorus pesticides like diazepam. As we continue to explore interactions between pesticides and microorganisms, researchers can gain deeper insights which can support agricultural productivity without compromising the health of our soils and waterways. Thank you for listening.